You're listening to The Interactome, a podcast by a group of young researchers who want to connect you to the world of science by sharing their stories and perspectives. Just in case their bosses are listening, they want to remind you that the opinions expressed here are their own. They also want to remind you not to take anything they say as medical or professional advice, as they are not doctors. Not yet, anyway. Stay tuned about that. And, without further ado, welcome to the Interactome.
Hi everyone, and welcome back to the Interactome. This is Natalie, and I'm joined by my co-hosts Sam and Joe. Here today, we have a guest-free episode, and we're going to talk about some really interesting stuff related to genetics. But to kick this off, Sam's going to tell a story about a guy who was mostly wrong one time.
Sorry. Yeah. What a way to start off.
Yeah, not unusual for me, though. If you dial it all the way back to our first episode, I talked about a guy who got famous for boiling his pee.
Good point.
So,
Good point.
In the same vein, we're going to talk about a guy who, way back when, actually was doing some very interesting scientific thinking in the turn of the 1800s. And if you know anything about biology in the 1800s, odds are, unless your name is Charles Darwin or maybe some other people who come up sometimes, you're probably wrong. Also, Darwin was probably wrong about some things too, and we're getting to that because it turns out Darwin actually kind of copied some of this guy's ideas. Science is an iterative process, and the person we're about to talk about is not the first person to think about evolution, but he is one of the first people you learn about in school who thought about evolution. Today I want to talk about Jean-Baptiste Lamarck, who was a scientist around the turn of the 1800s who was trying to figure out where life came from, how it changed, and how different traits were inherited. So this is, as a reminder, this is 70 years before the discovery of DNA, which at the time no one really took seriously, and I think about 60 years before Charles Darwin published The Origin of Species. So we don't know about DNA. The concept of evolution hasn't really hit the ground running. And so Mr. Lamarck was primarily trying to figure out, well, okay, so we have all these fossils. We have all these things that don't exist anymore. We have a bunch of life that is adapted for how this place is where it lives. And how does that happen? And he took a look around at people, and he also started asking, why, for example, a blacksmith is typically, someone who works in blacksmithing would be strong and stocky. Someone who has to run a lot would be relatively skinny and, good at running. And starts asking these questions, goes, well, why does that happen? And the conclusion that Lamarck reached was it was because their parents were good at it, and the reason that their offspring have the traits that their parents have was because their parents tried hard. Their parents did these things, and so therefore their children will be better at it. The kind of canonical description that everyone gives is that giraffes have long necks because they kept stretching their necks. They kept trying to reach higher and higher branches, and that was passed on to their offspring. And this is what Lamarck thought as part of his model of heredity and part of a much larger model of evolution that Darwin actually did pick up on. It's worth noting that Darwin even talked a little bit about this idea of inheriting acquired traits. It was not necessarily debunked until the early 1900s. But we know now that the majority, at least, of the traits that organisms have happen as a result of random mutations that just happen to be beneficial. If you're thinking about mutations, you may think about the Teenage Mutant Ninja Turtles, but you're probably also thinking about a horribly deformed frog with 5 legs and 3 eyes that lives in a toxic waste pond or something. If you've watched The Simpsons, there's a couple jokes about this, and the that is kind of a relatively true depiction of mutations. Most of them don't help, some of them do, and so it makes sense that someone like Lamarck, looking around at the natural world, would say, well, it's probably not the random stuff that happens that, makes a horribly stunted tree or something like that. It's going to be, trying hard. It's going to be, adapting to your environment. And up until recently, modern scientists were pretty sure this was completely wrong. But by the end of this episode, we'll talk about how that's not entirely true. But I think turn this over to Joe, who's more of an expert on today's subject than I am.
Oh boy! All right, let's see what I can do. Before we kind of like get into it, I just want to say this area is super super interesting to me personally, and it is something that really like kind of got me into science. I first learned about this. We'll get into it.
Joe, safe to say this is your villain origin story.
I was just gonna say you're a huge nerd. But
I villain nerd, maybe both. I don't know. It remains to be seen, I guess.
Most supervillains are nerds deep down.
That is a good point. I hope to use science only for good. But yeah. So I back in college in my first year, literally first semester, I walk into the classroom for my biology class with everyone else in biology, and we sit down, and the teacher, who was a little chaotic, walks in, and she's like, "All right, everyone, today we're going to learn about licking rats." And I was like, "Okay, what the heck?"
Oh,
and I was like,
"That's what I thought too. That's crazy."
All right, and so I was a little weirded out by it. Yeah.
Natalie, what did you think?
Oh, I was just being like, "Oh yeah, licking rats, duh."
Okay,
that's not what you thought, Sam.
Well, you two went to school together, so I don't know if Natalie's like, "Oh yeah, I've heard this story 1,000 times before."
Natalie and I took separate biology classes.
Okay. So, I knew.
Yeah.
Okay.
No worries at all. But
I like to think that if a professor ever said that to me, I'd remember though.
Yeah. So she started talking about this experiment that someone did, where basically you had some rats, some that licked their pups, and some that didn't. And in rat, if you're a rat, licking your pups is basically the rat equivalent of giving your kids hugs and kisses and caring for them and providing physical comfort. Especially in early childhood, we know that kids really need physical contact or emotional support and through gestures of care, and that's actually very important. Immediately when children are born, one of the first things that doctors always talk about is skin to skin contact. And so this is kind of a rat equivalent of this. And so the in this case they looked at rats who licked their pups and rats who licked their pups a lot less, and they actually saw that there were basically the rats who were not licked a lot would lick or would not lick their pups, and so they wouldn't basically the behavior was passed on, even though the behavior was really only happening very early on in development. But then what they did was they took the rats and they took the rats that were immediately born from a licking mother and a non-licking mother, and they switched them. And so you have rats that were just born from a mother who were licked, or who would normally lick her pups, who are going to a mother who doesn't lick her pups. And then you could see that the mother that they were raised by dictated how they behaved when they were older. And so that was super interesting to me. And we got into, and we'll talk about it, we got into some of the molecular mechanisms that may be behind this phenomenon. I think there's some debate about whether this specific study actually is replicable, but regardless, it was the study that got me super interested in the idea that, oh wow, maybe in some cases our environment or our experiences may actually influence behaviors and how our genes are used, things like that. What's up, Natalie?
So, Joe, how does that relate to the whole, I think, nature versus nurture argument that I'm sure a lot of listeners have heard of before?
That's a really good question. At least in this specific study, it seemed that the behavior was at least partially due to nurture in this case, which I find intriguing because in a lot of cases, like in the vast majority of cases that we know of, most traits or things are often due to nature. And like I think that behaviors themselves are very, very complex and like multifaceted, and like I'm sure a lot of neuroscientists would have thoughts about whether you can specifically say that one behavior is due to nature or nurture alone. But in this case, like they were able to prove that, at least in this one situation, nurture did play a role, which was fascinating.
How are we sure the rats aren't remembering it?
It's a good question. I don't think we can, honestly. And I actually, I'll get into how I would want to improve on this rat licking experiment later on, because I have some thoughts about how we can understand the molecular consequences of rat licking. And it's an experiment I've wanted to do like forever that I don't have the resources to do. But we'll get into that. But I think the like given like there are a lot of examples like these where like you see like a trait that like we're starting to kind of like come back to Lamarck maybe a little bit in some ways. Like, and we can see like in certain cases, like in this example, there was a behavior or a phenomenon that was kind of like passed down through at least a few generations. And so was he completely wrong? I think it's really like starting to be up for debate again, in many ways. It's very clear, like there are like most of our traits are genetically determined, but I think today it'd be good to talk about epigenetics, which is kind of like the thing that really determines how our genes are used. Yeah.
And the word epigenetics, doesn't that mean above the genome? Like on top of the genome?
Yeah. It's like kind of if you, I think a good way to think about it is like if your DNA is the hardware like is just kind of like your epigenetics are kind of like your software. They're like kind of like the program in the cell that kind of like tells the cell how it should be doing things. Like, should it be a brain cell or should it be a heart cell or things like that. And like it's actually like really, really, really complicated. And I think we're gonna get into a lot of it today. But I think the real answer is we're still learning a lot about it. Another good analogy could be like let's say you got your maybe your genetics are like your recipe book that has been passed down through the generations. Like in this case, this recipe doesn't encode or doesn't like tell you how to make cookies. It tells you how to make certain proteins that your body needs, like to build cells and mitochondria and like to build to basically make an organism. But your epigenetics are kind of like all the sticky notes that like you or like your grandma or whoever like put in there that are like, oh, actually like you need like double this recipe to get what you want, or like actually don't, this recipe's bad, don't use it at all. Yeah. And so like you will like as the person like making the recipes, you will like ignore certain recipes that are just like not good. And you might pay more attention to the ones that are like you've put a sticky note there like this one's really good. Like we need to make more of this. Yeah.
I guess this is a bit of a tangent because I know we're going to get into like the mechanisms of epigenetics, but why would, maybe just kind of taking a step back,
sure,
why would a recipe book have recipes that are bad to begin with? Why would you need those sticky notes to then kind of course correct?
Have you ever bought a recipe book, Natalie?
Honestly, no. I don't cook that much.
I've got like several recipe books, like cookbooks and things, and there's always like a couple of them like you do not make that one.
But I'm talking about, I guess,
in the context of biology.
Yes,
yeah.
that's
yeah. I think it's all context dependent. Like if I think a good comparison is like, I mean, there are certain foods that you don't eat for breakfast. There are certain foods that I personally don't eat for lunch, and some that I don't eat for dinner. I'm usually not. Sometimes I'm in the mood for having pancakes for dinner, but usually I'm not. Usually pancakes are like a morning kind of thing. And so, like, in response to the time of day, my environment, or like how I'm feeling, or like my mood, or things like that, like other stimuli or other people I'm with, I might want to make different recipes or eat or use like different, like kind of use different things, if that makes sense. And I think that's a good comparison, like a good analogy for like if you're like a generic cell hanging out with all the other cells and they're all sending you certain signals, like, oh my gosh, there's like inflammation happening, or like, oh wow, we just got a lot of sugar, woohoo, like like the things that you as the cell are going to make are going to be different. And that is it's a good example of how, like earlier we talked about an example where there's this trait that was kind of like passed down through multiple generations of licking rats. But this, like, and we'll get into the potential mechanisms of that. But in this case, like, this isn't like this kind of analogy is a good example of how epigenetics can be dynamic and respond to your environment too. Not just on the scale of generations, but in some cases, like over the span of having done some work in like this context, like actually like in the span of like hours, about, I'd say. Yeah.
And the other thing that is also the case is sometimes your conditions change completely. And to extend the cookbook analogy, I don't know if y'all have heard of a savory jello salad before.
Wait, what?
Yeah, so they used to make like jello molds. Like this was like all the rage in like the 70s and 80s. Actually going back, I think even to the 50s when like jello started becoming a thing. And like they'd put like tuna in jello and serve that as an entree.
Wait. This happens every Thanksgiving in my family.
Well, yeah, but the thing is, it's also context dependent too. So maybe in your family, it's not entirely disadvantageous to have the trait to be reading off that recipe, because at least everyone understands what's going on. But like for
nobody understands what's going on though. Like no.
Who makes it?
My grandmother. So I think it's like there are certain people who love it, but it's like in this case, it's like orange jello with like ricotta cheese.
Oh yeah. I've had that before. And, I'm talking like nasty stuff. Like I'm talking like celery, tuna, like. We're talking like you take straight gelatin, you start putting like cottage cheese. It's not, nobody, I hear it's maybe popular in the Midwest, and I'm sure like some people are like, I haven't. Okay. All right. Well, if you're not in, whatever. But the point is, some things are useful in certain contexts for the analogy. There's some things that were like at the time that was an advantageous thing. It's like, oh, everyone's making jello, I'm going to bring this jello thing. But like now it's like, you don't make that recipe anymore. There's going to be proteins, like there's going to be genes that like, don't make sense in the context of even necessarily, possibly even a human. But at least in the context of you now, you know, those genes might have been useful for ancestors, but now they're turned off.
Nana, if you're listening, I like all of your cooking. I promise. But
yeah. I'm not talking about the orange jello with like some other stuff. I'm talking like There's some people on the internet. It's like I saw one recently that was like it's like coleslaw but in gelatin, but it wasn't even like a good coleslaw. It involved pureed cottage cheese and like it was like a whole thing. Apparently, it was not good.
I'm gonna be honest, Nana, if you're listening, I like all of your cooking except for the jello, and I'm sorry.
Okay, but like we have that a lot. We're so lost right now. I was like, I got a great laser focus here of something that was relevant in a past generation that's not now, and we've marked that gene. We've marked that as like, we don't make this now because it doesn't make sense for the context. Which, you know, we could talk about like gene silencing, but
we'll get into
I'm sorry, Joe's Nana. I did not mean to do this to you.
I think she's a good sport.
Okay.
But yeah, no, it's. I think these are all like really, really good comparisons, and like these phenomena like do really happen in at least in animal models. It's been somewhat controversial to demonstrate at least some of these intergenerational effects in humans, but like we can see like that in or in a lot of contexts, like at least the dynamic epigenetic regulation, like responding to your environment, does happen in humans for sure. And so that's yeah, it's really, really fascinating. I don't know if we want to get into the mechanisms now, kind of like how does
Let's
it
do
work?
it.
I think the biggest one, and this one's actually kind of relevant to the rat licking, is DNA modifications. So like, you know, like how people are like, oh my gosh, like they like if you do this thing, it's gonna like change your DNA and stuff like that. And like people worry about like certain things changing their DNA, and to be honest, like that's like the only things that are really changing like the actual sequence of your DNA are mutagens. So like things like carcinogens that like cause cancer and stuff like that in a lot of cases. But
And the sun.
And the
just
sun
cause cancer.
Yeah.
Just cause cancer. It's a mutagen.
Yeah,
But,
absolutely. And it does literally
Reyes.
change the structure of your DNA.
Yeah.
That's how it causes. It literally gets two bases in your DNA to like stick together in a bad way.
Yeah, so I guess with that, I mean, when you talk about carcinogens, you're talking about actual mutations. So
Yes.
the epigenetic DNA modifications then are not mutations.
No, they are not. And I think this is really, really interesting. Like you can, in this way, like basically you can have your DNA with all the little rungs in the ladder. And it's kind of like putting like if I'm climbing along a ladder, like let's say I'm a protein that's like reading the DNA, going along the ladder. I might kind of like chug along the ladder until I see a note kind of like stuck to the side of the ladder. And then I can just kind of like grab the note and look at the note. And if it says like, hey, stop here, don't climb any higher, like I'll just stop. And like that's kind of a good analogy for DNA modifications. They're kind of like little bits of chemical like that get added to kind of the DNA without changing the sequence itself, which is really, really fascinating. The most common one is DNA methylation, where a methyl group in this context is a kind of like it's one carbon atom with 3 hydrogen atoms attached to it that's then popped onto one of the rungs in the ladder of your DNA. And usually areas that have lots of these methyl groups on them are considered to be silent, as Sam was talking about earlier. Like basically like the cell is not reading that information that is in that part of your DNA. Like they're not it's like it would be like a sticky note that says don't read this part. Don't look at this part of the book.
Don't make the jello salad.
Don't make the jello salad.
And then sometimes too, I guess what I'll add is that sometimes like you may be thinking, you listener, maybe thinking like, oh, well, why would you have DNA that like didn't need to be read? A lot of that too is like and maybe this isn't for all of it epigenetically, but there are these things called like fossilized DNAs or fossil DNA.
Oh my
where
gosh,
it's like,
I totally forgot about that. Yeah, sorry. It's exciting stuff.
No,
Yeah.
it's okay. So it's like this, these, DNA that was valuable when, when like there were dinosaurs or like when we were, not necessarily Homo sapiens yet, but like instead of it's less intense for, I would say, like the body and the cell to like completely cut out and like get rid of that DNA, because it could by proxy get rid of some really important information in the DNA. So it's easier to just like shut it off like a light switch.
Yeah.
Like just kind of like, oh, tag this on here, and then, it's less intensive for the body and for cell replication to just get rid of it. So then that epigenetic trait gets passed down.
Yeah.
To extend the jello salad metaphor even further, because I'm enjoying this too much, it's like you don't rip the whole page out because your cousin's birthday cake's on the other side of the page. You just put a sticky note there, like, we're not making jello salad anymore. It's, not 1960.
That's a great
What
example.
I'm so impressed by is that We didn't prep these jello analogies
No.
ahead of time.
No.
Sam is just off the cuff
This,
bringing them.
This is what the Interactome does best.
No,
So yeah, improv, off the cuff, and yet it's better than what we planned in the first place.
Science improv. That's what this podcast is.
Oh yeah. Kind of. I want to build off that though, because that is actually Natalie, what you're getting at. Like I've like talking to some of the epigenetics folks where I research, like some of them think that the reason that DNA methylation actually evolved in the first place is really to do that, to like shut down the parts of DNA that we don't really want anymore. And important thing to note is that it's not just like genes that were used that we're not using anymore in a lot of cases. In a lot of cases, it's actually viruses that have integrated into our DNA, like going all the way back to our first, like, what is a virus video? Like retroviruses that get into your DNA and like stick themselves there forever. The cell can actually just kind of like put a sticky note over them and just kind of like say, shh, ignore this, so that your cells don't make more virus. And like that happens over the course of evolution, like over the millions of years, not necessarily like immediately. What's up, Natalie? Did you have a question?
I guess, and maybe we're gonna get into this, but you know, your virus example kind of triggered this thought.
Yeah.
How does the body know, or how do cells know where to put these methyl groups or these little sticky notes for these tags? And maybe this is something, Joe, your eyebrows are raising, so maybe this is something we're gonna get into.
We're gonna get into it, all right. And I think it's an area that's one, hotly debated, and two, extremely complex, and the short answer is we don't know everything. The long answer is we know some stuff, but not all. And so the one thing to note is that these DNA methylation modifications aren't the only epigenetic mechanism happening in a cell. And so they're acting in concert with a lot of other weird mechanisms. And so one of those is histone modifications. So histones are kind of like spools that your DNA is wrapped around. And the reason cells do that is to kind of like get everything to fit inside the nucleus. Because like if you stretch out your DNA like in one big strand, it's super, super long, like way wider than the width of a cell. And so like you kind of need to scrunch it up in order to get it like to fit in the cell. And so in this case like there are actually little chemical tags that can get added to the spools that things are wrapped around. And so those tags can say, hey, we're going to unwind some of this DNA, or we're going to wind up some of this DNA. And like those tags act in concert with like basically those tags actually in many cases can like serve as markers to recruit proteins that then add the DNA methylation, which is really like it's like oftentimes histone, at least to my understanding, and don't get me wrong, I'm just starting my PhD. Like those modifications on the spools actually are kind of like little signals in a lot of cases that say like, hey, we're gonna like add or take off certain DNA markers, which like I found that fascinating. And the other thing is like those histone markers are oftentimes working by themselves, or sometimes like the other thing to note is that another really, really interesting mechanism that I think here where I research, like I got to dig into it a little bit, is how the DNA is actually scrunched up matters. Like not just at the level of the spool, but the level of the whole jumble of DNA. Like, and so like, let's say you have a lot of different loops of DNA all looped together, like how all those loops actually touch each other, and like how, like what loops like a given loop are next to, like those actually matter. Like we didn't think about that for a long time. We just thought like the sequence was what matters, but literally how the DNA is jumbled up in 3D space can play a role in what genes are used in a cell. For example, there are like in a lot of cases, like to determine the identity of a cell. Let's say like if you're comparing a heart cell to a brain cell or things like that. A lot of the genes, like the genes that make or tell the cell to be a heart cell, will be like kind of shoved close to the edge of a like the nucleus where all the DNA is in a cell, and vice versa in the brain. So like the brain genes will be closer to the center of the nucleus in a brain cell, and in a heart cell, all the brain genes will be closer to the edge of the nucleus.
But then are the heart genes towards the middle of the nucleus for the heart cell?
Yes. So yeah, it's kind of like it's a bit of a simplification, but the in a lot of cases, and this is something that we're just starting to learn the mechanisms of, the distance that a given segment of DNA is from the periphery, like the outside or the edge of the nucleus where all the DNA fits in a cell, like kind of like the sphere that the DNA is inside, can determine whether that gene is used or not. And so like that is like a lot of the genes that are like literally stuck to the edge or to the inside of this sphere, which is the nucleus, are all turned off. They all have like histone modifications that say don't use me. They have DNA modifications that say don't use me, and the ones closer to the center have kind of like the opposite modifications. So it's a really novel area of research that people are just starting to get into. There's all these crazy sequencing technologies where you can like basically like stick a bunch of the DNA together and like cross-link it, where you basically force things to attach, and then you chop them all up and then sequence them and see what's stuck to what.
And I think one thing that's interesting, at least to me, about that is that when we talked about epigenetics at the top of the episode, we talked about behavior, right? And how behavior can be inherited through, environment. Yeah. It just shows that epigenetics is so broad and it covers such a wide umbrella, because like that is not those locations is not determined by like your parental figures. Like it's an inherited. Yeah, I'm kind of losing.
It's like a biological trait. And so like in this case, I think it's a good example of how epigenetics is kind of like a broad phenomenon that mediates interactions between both nature and nurture. And so like I think that's a good way to frame it in a lot of cases. Like these are not all, but a good portion of mechanisms that mediate the effects of both the genes and the environment.
See, this makes like a weird intuitive sense to me.
Yeah.
In the sense that like, if you've ever like gone to a library or dug through a database or something, depending on who's going to be using the books or who's going to be reading those documents, it's going to be organized differently. It could have exactly the same information in it. Yeah. But based on like, it's weird how like it feels like very concrete and like a very like human sense of like, if I want to organize information, I'm going to put the stuff I use somewhere where I can get to it. But like if I use this never, like, someone might walk into the library asking for this obscure book on, underwater basket weaving from like, 1963. And like, we're going to keep it down in the archives, and you got to go look it up, and you got to go, okay, well, where is this? And it's like, that makes intuitive sense to me.
No, that's exactly it. Like, literally, like, this is very novel, but there are some studies that now suggest that, like, genes, there are certain things in the nucleus called nuclear speckles. And this is where, I think we talked about it in a previous episode, but RNA splicing happens, where you're like, the genes are making RNA, and then the RNA is getting chopped up into different segments because only certain segments of that RNA is getting used. And genes that are being transcribed and then spliced tend to be closer to nuclear speckles, whereas genes that are not tend to be a little farther away. Which is, I think, really, really interesting. It's exactly a molecular mechanism that exactly represents what you're talking about.
and I think, like, to me, like, this, so, okay, so now I start talking about these things, people are like, well, but this evolved, right? Like, this is not a thing that was made, you know,
No.
that's our understanding. And so, why does this happen? But then you can go, well, if your library is a mess, you're gonna be less efficient, you're less capable of doing something. It's like any improvement to the organization would be beneficial, because that means genes are transcribed more quickly. The cell can respond faster. It's more likely to survive.
Yeah.
It's the path of least resistance.
Yeah.
I guess, been having that organization that you accumulate over time.
Yeah. That's a really good point. And did we just come up with a new theory of evolution? I don't know. But so
Jello.
hello, the Jello theory.
The jello salad theory of evolution.
Yeah, no, it really, like, but I think it's a really good point. Like, it's a huge, significant point about, like, why genome regulation evolved in the first place. And the other thing that I'll add is that, like, we're talking a little bit about RNA, but RNA, like, we don't normally talk about, like, DNA goes to RNA, goes to protein, but, like, RNA actually may be one of the mechanisms that plays a role in a lot of cases, like, orchestrating all these different, like, tags and stuff like that. So, yeah.
I mean, I spent 6 months working on characterizing a protein that does histone methylation.
Yeah.
And we were looking at proteins that interact with the protein that does histone methylation.
Yeah.
So this is like, so you have the DNA structure, then you have the methylation, which is regulated by these histone proteins. And the histone proteins are what the DNA is wrapped around. But the histone proteins are modified by other proteins, which are in turn controlled by other proteins, which probably, it's like, if you've ever looked at a map of how the inside of the cell works, it looks like somebody started tacking protein names onto a bowl of spaghetti that they dumped out on a table. And that's why, is because it's this hugely complicated system. Like any complex system. I mean, even the ones that humans build, they're less complicated, but they're still, really, really complex. And it's like, yeah, so you're talking like RNA influences this, but also there's other proteins. And like, there's these, like, the proteins that are working, these little tiny disordered proteins that did nothing unless they stuck to the protein methyltransferase that was modifying the histones. And it's like, that was a wild thing. It was an internship.
Oh yeah.
I think I was the first person to purify one of those proteins. It was literally the boringest protein I've ever purified. But
I'm so sorry.
No, it was great. It was great. It was actually, the context of that's a whole nother story.
Yeah.
But it was actually good that it was boring, because there were other issues that were happening that had nothing to do with me. I was like, oh great, something worked.
Yeah.
But that's tangents on tangents,
Well, kind of building off of that tangent in a way that brings us back. I think it's a good example. Like, it's a perfect use of the "It's Sunny in Philadelphia" meme, where you see like the guy with like the crazed dude with the map,
Charlie.
Like, yeah. So I think of that.
That's the paper on the cork board with all the strings and everything.
Yeah,
I mean, it's the Pepe Silva.
That's the cell, but the cell is like infinitely more complex. And one thing that I think is fascinating, like, kind of talking about the protein stuff, Sam, there was actually a paper that was just published that claims that at least in worms there was a specific, like, amyloid protein, like a protein kind of like clumped up, that would be passed down from generation to generation and control the sex of the worms. And so essentially, like, you passed down this protein, and it had a tendency to make the worms female. Like, normally the worms, like, every time they reproduce, switch between male and female, but they were tending to be more female as time went on, like, throughout the generations. And, like, I think this will have to be replicated, but the researchers, like, they were getting weirded out because all their worms were, like, becoming female, and, like, normally you have, like, 50% male, 50% female, and, like, but they thought this phenomenon was so cool and so interesting that they dropped what they were doing to research this, and, like, 10 years later, this is what they found. Go ahead, Sam.
I was saying, and to back up, like, I think the audience is probably familiar with, like, how sex is determined in humans, where it's like, you have sex chromosomes, and most people who are female have XX chromosomes. Most people who are, biologically male have XY chromosomes. That's not always true. Even in humans. when you get past humans, you get, especially outside of like mammals, it becomes just
Weird.
there's like turtles, it depends on the temperature. There's some fish where it's like there's only one female or something, and like, or one male, and it's like one dies, the rest. Sex determination in biology is insanely complicated. It is not locked down. This is not shocking that there's a protein that makes worms female.
Yeah. It doesn't mean that all your kids are gonna be one biological sex or things like that. So don't worry about that.
There could be something in humans that messes with that. I mean, you could copy a bunch of. Yeah, there's definitely. There's plenty of weird things that happen with humans too, and we don't talk about that enough. But that's like just for context. It's not like, I don't know how worm sex is determined. I don't know anything about these worms. You'd have to look at that worm in particular. I'm sure there's varieties among worms.
Yeah. I kind of want to get back to the rat licking.
And your licking
take
rats.
it away.
So, we were talking about rat licking. We didn't really get into the mechanism of it, but the thing that really, really drew me to epigenetics was this claim that they, like, basically they did some, like, sequencing analysis and saw that there was, in the mice that were not licked by, or the rats that were not licked by their mothers, they had increased levels of DNA methylation. So a DNA modification that tends to shut down genes. And in the specific gene that they looked at that had increased levels of DNA methylation was the glucocorticoid receptor gene. Essentially, this gene is the gene that tells your body when it's made enough cortisol, the stress hormone. Like, it senses the your stress hormones, and essentially, like, its job in the brain is to, in a lot of, it does a lot of different things, but one of its main jobs is to sense when you've made enough stress hormone and tell your brain to stop telling the rest of your body to make stress hormone. And in the mice that were like not licked by their mothers, they had higher levels of stress hormone, in large part, in the opinion of the authors, because they didn't have as much of this receptor to see that there was hormone in the first place. And so then that is, does someone have a...
I have a question, just because I guess, and I'm not, I guess, like, I don't want to come out as I'm, like, insulting this. I haven't read this paper.
There are I will, like, there are people have tried to repeat this work and it hasn't always turned out well. So
it sounds a lot kind of like causation, or correlation as opposed to causation. Yes. But my question was, you mentioned that later, or there was a different study that swapped the rats.
It was the same study. Yeah.
It was the same study. So then, how did those cortisol levels get affected?
They switched, essentially.
They switched.
Yeah.
Okay.
So the authors claimed that the level of DNA methylation and, like, the cortisol, all that stuff, like, correlated with how the mother treated the baby rat early in life. And some people claim that, like, certain, like, traumatic experiences and things like that can be passed down epigenetically through generations in humans. And there are some studies that do suggest that may be the case. But, like, there are other studies that refute that. I think one thing that I would love to do long term is to kind of repeat this rat licking experiment. Except what we could do is we could take the you could take we talked about this a while ago, if I remember correctly, CRISPR gene editing. And you could take a special version of this CRISPR gene editor that doesn't actually cut DNA. You have a special protein attached to it that removes DNA methylation or adds DNA methylation. And so you direct it to go to this glucocorticoid receptor gene and see if messing with the DNA methylation at that locus, that region of DNA, abolishes or stops the effect of the whether the mother licked the rat the baby rat or not. And like that would be the ultimate way to really test whether this whether this is correlative or whether it's causative. And so that's an experiment that I've wanted to do for a very, very long time and have just never had the resources to do it. So any listeners, if you want to do it, go for it. But
you're just giving out your ideas.
Hey, I got plenty. I'll be all right. So yeah. But like in all seriousness, like that is like we're getting it to the point where we have like technologies to start manipulating the epigenome, and like this would be a really good experiment to test to see like if some of these things really are real in the way that we thought. At the bare minimum, there are some studies that actually show that like I know we talked about RNAs, but like in worms, you can basically like that people have demonstrated that there are certain little bits of RNA in the sperm of worms that actually control like basically if you change the types of RNAs, or if in the sperm of a male worm, it can influence how genes are used in its offspring. And then how those genes are used can like stay the same for multiple generations down the line. And they've actually like basically like taken the RNAs out of one sperm and then put them into another sperm and like changed it all around. And like it's very, very clear that at least for the RNAs, like those actually mediate an intergenerational phenomenon, which is fascinating in worms. So yeah.
But where do the RNAs come from? Don't they come from the genome to begin with?
They do, which is the other thing. But those RNAs were the types of RNAs that were present were also partially dependent on like certain stimuli, whether like a certain like odor or stimulant or things like that. And like you could actually take that and like that specific those RNAs that were like where you have like an exposure to a chemical or things like that and put it in like basically the sperm of another worm and like have the same effect.
So essentially the worm smells something. It expresses a gene to make an RNA. Mm-hmm. That RNA then ends up in its sperm, which then influences its offspring. But you can also just take the RNA and then stick that into a different worm, and it behaves like its father had smelled something.
Yes.
Okay.
Yeah.
Just working through that.
Yeah. And so like I think like I'll need to like and I'll fact check this myself, and we can like post like this just to double check. But like I've actually worked with people who like do this stuff. Like I don't know if the smelling is a specific example that's true, but there are like people have done this, and it's really cool.
He who smelt it has different offspring than he who did not smell it.
I guess so. Yeah, it's just
that's wild.
It's like it's fascinating stuff, and we're only just starting to learn about it. So I don't know. Go ahead, Sam. Sorry.
So I was gonna say, so if I smell certain things, are my kids gonna be different?
I don't know.
Like, how does this translate now? So, like, why does this matter? Like, okay, we got worms smelling things, we got rats getting licked. Like, how does this, translate to humans? Or go back to Lamarck and his stretchy giraffes.
Yeah. Well, I think there are a few different ways. First, like the most easy answer is that there are certain genes, like certain cancer genes that if you mess with them, they cause cancer. And a lot of those genes are actually genes that control the epigenome. And so there are certain anti-cancer treatments that target your epigenetics in a way that, like, because of the way the epigenetics are changed in your cancer cells versus the human cells, you can specifically kill the cancer cell. So that's one very, like, clear, like, very direct example that's relevant for human health. However,
I think that was what we were trying to do with that protein I was working on. I mean, like, I was an intern, so I didn't quite grasp this, but there was some oncology implication. There was some pharmaceutical company that we were talking to. This is all, like, way in the past. It's published now, but, like, there was, like, this whole thing. It's like, okay, I was like, what? I didn't quite follow. I was a little biochemist who was very excited to have a bunch of shiny toys to play with.
Yeah.
But yeah, that tracks with the very little I know about.
Mm-hmm.
So the protein I was working on was, I guess, probably one of those genes that controls the epigenome.
Yeah.
So that makes sense.
Yeah. The other thing is that, like, I think a good example of this is, like, twins. Like, they're genetically the same, but
identical twins.
Yeah, identical twins. Good point. There's also the like non-identical or fraternal twins, but genetically identical twins. Like one can like go on to get a disease that the other doesn't, things like that, even though they have the same DNA. Like one may behave a little differently, even though like than the other, even though they have the same DNA. And like I have friends who are like twins, and like multiple pairs of friends who are twins, and like sometimes they want to like be different than each other, because like when everyone's kind of assuming you're the same, that's can be hard sometimes. And so like I think it's reassuring to know that like our DNA isn't the only thing that makes us who we are. Like it makes up a lot of who we are, but your epigenetics play a big role too. They're kind of like mediating both the nature and the nurture. And I think in a way that's kind of liberating for a lot of people, from a philosophical perspective. So yeah, that's my thought on it.
And we've tested this too, right? Like there's been some studies. There was an interesting one where there was a long-term astronaut study where two identical twins, one's an astronaut, one's not. And the astronaut one, I think, stayed on the International Space Station for over a year, came back, and I believe his epigenome was a little bit different.
Yeah, specifically they looked at DNA methylation, and the DNA methylation patterns were like completely changed. Or not completely, but significantly changed.
So I guess if you're identical twins, you want to be a little different. Just one, you just got to spend a bunch of time in low Earth orbit, which is easy enough.
Heck yeah, let's do it. Yeah, man, what a wild area. Like I could,
and we just, like you said, Joe, we only just scratched like the very, very top of the surface.
Yeah.
Like this stuff that goes on with this stuff is, and for listeners, I've known Joe for, I don't know, since we were a sophomore in college, and I still remember for Joe's first thing was like, yeah, I'm all about epigenetics. Like I'm gonna research epigenetics.
Yeah.
Like that was like, that's been his. So I've been begging the team to do this episode for a while, and I'm sure that we're gonna come back with like a part 2 or part 15, because there's so much to literally go into.
Eventually Dr. Joe will be on here talking about his cool research and how your rats are licking each other or whatever.
Yeah. Well, we're it's kind of weird because I'm like, I'm in a genetics and epigenetics PhD program, but I'm doing metabolism research. So it's kind of like, it's a little weird that I think personally, like I've pivoted a little bit, but I would love to eventually kind of talk about kind of metabolism and epigenetics, how the two relate, like how, like eventually, like, I mean, like the epigenetics is relevant to like what happens as we age too. And so like your epigenome changes as you get older. And so we're just starting to uncover a lot of that too. And that's something I'm really fascinated in.
Yeah, you've remained remarkably focused over the time that we've been doing Interactome, at least, Joe. I mean, I think I've changed my career twice, and you're like epigenome. So I guess there's something there for you. I mean, I've never really worked with it. Maybe I would be that focused if I got into that.
I hope so. But yeah, it's interesting stuff. I think for me though, personally, like I'm only interested in it. Like I learned this the hard way, honestly, that I'm personally only interested in like studying epigenetics when it is directly relevant to human health. Like if I'm just looking at how the technical like mechanisms of how things work, just to know how things work, like it's cool, but it's not sufficiently motivating for me personally, on a personal level. Like I actually had a conversation with a good mentor of mine whose lab I was working in doing this stuff, and he said like, hey, like you're good at this stuff, but you need to do something that's directly relevant to human health. And like that was a really powerful and important mentorship experience, because he was explicitly, like, not in a bad way, but like telling me to go to a different lab because he knew that, like, he understood what motivated me. And like I hadn't fully understood it yet. And I know, like, now being in a different lab where things are very relevant to human health, like, I know that he was right. But like I want to get back to like the epigenome, and really like tie it to disease and aging and all that stuff. So yeah.
Well, thanks for taking the start to just talk to us about it today. I think this was a really great episode, and we covered a lot of ground. And Sam, I don't know if there's anything else you want to add.
Oh, I was just gonna say I don't think we talked about Joe's fascination with aging research very much either.
We'll get into it.
That's a whole
that could be a series. Which like we're gonna do it eventually. So I for our listeners, I'm thinking a lot about a metabolism episode and a biology of aging episode. Maybe multiple, because like all this stuff is related. So maybe a little preview for the future, if you will.
Hey Joe, if you're real successful, we'll live forever and we'll have all the time in the world.
well, there's a lot of we'll get into that in a future episode.
Well, thanks everyone for tuning in. You can find us on Instagram at Interactome Media, and then on Twitter at _the_Interactome, and we'll see you next time.
Woo! Yay!
Thank you.
Thank you, guys.